Welcome to our experiment about fluorescence and absorbance by group 7B. Our goal was to identify the type of dye in a sample and the concentration using a spectrometer. How do we do this? Using spectroscopy. Each known and existing element has its own special light spectrum that spectroscopists use to compare spectrums to identify samples. The results are that we learned that our unknown dye was the Swatelli dye based off of the absorbance spectra that was produced and where the peak was located in relations to the wavelength. We also learned about how the absorbance spectra represents the wavelengths that were absorbed and the fluorescence data represents the wavelengths that were emitted by a substance. We used two different spectrums for this lab. We used an absorption spectra and a fluorescence spectra. There are two very large differences. So the absorbance measures how much light was absorbed by a sample. It is the most globally used method, and it is used to fingerprint substances and to measure the concentration of a molecule in a solution. The minimum absorption wavelength of a substance is the color emitted / what can be seen with the naked eye. Fluorescence, on the other hand, measures the emission of light produced when a sample's electronic states are excited. It's used in plane crashes. So when a plane crashes, fluorescein is released to allow for large sea areas to be dyed to aid in rescue so that the plane can then be found. Fluorescence doesn't always perfectly represent the wavelengths that are reflected due to outside sources of light and other variables. For example, fluorescence being related to the reflected wavelength is modeled by chemical substances, but not necessarily for biological substances. For chlorophyll, the fluorescence is red and not the green that we would expect to see. We use two models to help us understand this information. First, we use Beer's law. This explains that the amount of light that is absorbed is proportional to the concentration. Here you can see that we have a molar attenuation constant. We also need the solution concentration, and we also need the length of the path that is traveled by the light to help us find the absorbance. We also used another model, which you can see here on the right. As you can see, an excitation photon will come in and it will excite an electron that's at the ground state energy. It will then jump up to an excited state energy with a certain wavelength. When the electron then falls back down to the ground state energy, it will emit a photon. It will fall back down at a smaller wavelength than when it went up to the excited state energy. The wavelength changes from entrance to exit due to energy transfer to the involved particles. The excitation photon can transfer energy through heat loss and to the electrons in a kinetic form. This will cause vibrations and harmonic oscillations, which can build up to change the excitation state.
Here we have listed steps out for our experiment. We use a spectrometer, flashlight, water, several dyes and cuvettes to examine and learn more about the chemical substance absorptions and fluorescence. The distance between the flashlight and the spectrometer probe was kept constant throughout the experiment to minimize errors when comparing data. When obtaining the fluorescence data, it is critical that the spectrometer is placed 90 degrees angle of the flashlight. This helps filter light wavelengths being emitted, lowering the intensity readings to comparable values, and avoid light contamination from outside sources. It is also really important to make sure that the probe is close to the cuvette as possible to limit contamination from outside light sources.
These graphs help to illustrate how we came to the conclusion of our unknown dye being the Swatelli dye. Figure 3 represents the raw collection data from the spectrometer. We use this to calculate the absorbance of the unknown by subtracting the intensity produced by the water, source 2, by the intensity of the unknown dye, source 4. This created the absorbance spectra in figure 1. When comparing the wavelength associated with the peak, we concluded that our dye was the Swatelli dye. Note, due to the external light and other sources of error, the intensity values may be off, but the peaks should have formed at the same wavelengths. These graphs pertain to the fluorescence dye. The absorbance of the dye is shown in figure 1. The fluorescence was taken by using the previously described procedure and is shown as source 5 on figure 2. Figure 3 compared the fluorescein absorbance, the blue line, and the fluorescence, orange line. While the intensity values are off compared to the given graph of the lab handout due to errors such as the external light source, the peaks still occur in the same location. The peak of the orange line shows that green light is reflected through fluorescence, and the peak of the blue line indicates that the main wavelength that is absorbed is blue light, with a slight overlap of green light. This overlap can also be accounted for from the error of the external light source. Here we have the calculation for determining the molar concentration of our dye. Since we determined that our dye was the Swatelli dye, we used the given molar absorbance constant and plugged that into Beer's law. To determine the absorbance to calculate for the concentration, we took the log of the peak water intensity over the peak dye absorbance that was calculated. Throughout this lab, we were able to analyze absorbance and fluorescence in comparison to chemical dyes, and this has many real-world implications. We now can understand on a more physical level how we see different colors. A major source of error throughout all of our trials was the contamination of a light source, which was the classroom lights being on. There were other errors that could have occurred throughout the experiment, however, this is the main one that we would like to point out. For a future experiment, we could change the color of light that we use for transmission through dyes and analyze the absorption spectrum and fluorescence.